So now it's time for our keynote session. And for this keynote session, I'm your chair, and this session would have as moderator Les Bissiker. He will be monitoring the chat as well, because we have more than 300 participants online where your questions can be already posted. So if you don't want to forget question, every time you have something in mind, you can already post it. We are monitoring the chat. So for this introductory session of the 7th Curating Clinical Genomes meeting, we have brought to you someone that I personally admire, and he's a trailblazer for genomic research in Africa and African researchers in this field of medical genomics. I'm talking about Professor Ambroise Wonkam. Professor Ambroise Wonkam is a professor of genetic medicine and the director of Massachusetts Nathan's Institutes and the Department of Genetic Medicine at Johns Hopkins University in the USA. He is also the director of GeneMap, which stands for Genetic Medicine of African Populations. And until recently, he was the Deputy Dean for Research at the University of Cape Town in South Africa. Professor Ambroise Wonkam was born, raised, and trained as a MD in Cameroon, where he obtained an MD degree at the University of Yaoundé before he moved to Geneva in Switzerland for a training in cell biology. He moved from Europe to come back again in Africa to obtain a PhD in human genetics in Cape Town before returning again to Geneva to finish the specialization in human genetics. And his research is well illustrated by more than 200 publications in peer-reviewed journals and supported by more or about $20 million of grants obtained from the NIH and the Wellcome Trust. And this research especially focuses on the African variations, how that could impact health. And he is using as model sickle cell anemia, hearing impairments, and he also helped a lot to advance the ethical practices of genomic research in Africa, and especially how we approach the way we return incidental findings in genomic research when it comes to African research participants. Those researchers have been conducted across multiple countries in Africa, and he is the president, actually the current president of the African Society for Human Genetics. Most of you might have heard about Professor Wonkam when he discussed the 3 million African Genome Project, this new idea that would allow us to reach a place where we would start talking about equity or representation of the diversity among public resources. So Professor Wonkam, we look forward to be not only entertained, but also educated by your talk today, which is entitled Harnessing Our Common African Genomic Variation to Improve Health Globally. Professor Wonkam, the mic is yours.
So, merci beaucoup, Aimé, pour ton introduction. And I'm very grateful for your kind words. It's my privilege and an honor to be here. I'm grateful to Aimé, to Les, to invite me for this presentation. And it's good to have Africa at the center of the agenda of this meeting. I hope I'll convince you that we should all invest in African genomic variation in the next 10 and 20 years. So, I would like to, can I advance the slides, please? I would like to think that I'm a global citizen, having work and live on the 3 different hemispheres and doing the same job in 3 different institutions. So, and I actually gave the privilege actually to have comparison. Today, I will start with my early journey in Africa, and specifically in Cameroon. I don't think this thing is working well, my friend, up there. Is it working now? So when we started about 20 years ago a genetic medicine service in Yaoundé, Cameroon, we thought that introducing genetic diagnostic for prevention would be the best way of getting genetic medicine in. Because sickle cell disease was the most common genetic condition in that country, and is still the most common genetic condition of humankind. We started with the genetic diagnostic for sickle cell disease as a way of introducing preventing genomics. This child is actually the first one in which we did genetic diagnostic for sickle cell disease before birth in Yaoundé. That was about 15 years ago. And we did that successfully. I did actually the diagnostic with my own hand in my kitchen in Yaoundé, because I did not have the lab at the time, but I was so willing to show that it can work. And I set the lab in my kitchen, and it actually works. And we moved from there to set a formal clinic, moving from prenatal diagnostic to dysmorphology, describe many conditions that was known in the literature, but this time in the context of Africa, and also bring the notion that the concept of dysmorphology cannot be universal. For example, for this condition called Williams syndrome, if you have a child with European ancestry, like that little boy on the right-hand side of the screen, one of the clinical description is full lips. But if you look at all the other African kids, they all have full lips, like me, like anyone else in Africa, actually. So we advanced the idea that perhaps we shouldn't use the description that I learned when I was training, but the behavioral of this, the behavior of this kid, and also the malformation in the heart that they will all have. We move from dysmorphology for single gene disorder, for example, affecting intellectual disability, like this large family affected by Fragile X syndrome in the western part of Cameroon. One of the multidisciplinary clinics we set in Yaoundé at the time was disorder of urogenital malformation. When we started the clinic at the time, we did not have a lab, I said it before, but we did not also have the resources to provide proper treatment. So I went back again to Geneva and invited a few friends, some of which came from France, and we set this clinic that allowed us to have combined genetics, surgery, hormonology, and also proper phenotyping to help this kid. This, for example, a summary of the case that we saw the first 2 years. We were able to have a precise molecular characterization, for example, for congenital adrenal hyperplasia, and have a map of a country where all the mutations were located. For example, for this child who has that condition, we're able to provide a proper surgical treatment, and this can not only be done if you have a proper molecular diagnostic and have a proper counseling. From time to time, we will have a very complex case, like for this boy, self-identified as a boy despite the chromosomal composition of a female, and at the time we saw him, he was 14 and he had some breast development, and after the appropriate counseling, care, legal, he received a mastectomy and the proper hormonology treatment to grow as he wanted to grow. And based on that experience from Yaoundé, we were very confident when we wrote this opinion piece after the debacle of Caster Semenya that we cannot use genetic for gender testing, because I don't think even today we are educated enough to do that. Of course, if the American Association of Genetic Counselors is correct, this paper was the one used by the opponent of using genetic for gender testing, actually allowed the International Federation to stop doing that practice. If this is correct, that means that this simple commentary paper is probably still one of my most influential papers. So this gentleman on the right-hand side, his name is Nep. He's my cousin, but I don't think you know that. So I have some Nubian ancestry, so this guy could have been my cousin 4,000 years ago. So this gentleman had achondroplasia, as you can see. He has a beautiful wife with the normal size, and they had 2 kids. This gentleman was the chief of the wardrobe of the pharaoh at the time. He was also a priest, and toward the end of his life, he became a god. And he was, of course, buried under a pyramid because of his social status. And the reason I'm telling the story is because perhaps today we do not have the same tolerance for disability. We don't have the same tolerance for difference that perhaps we had 4,000 years ago. So when after a couple of years in Cameroon, I moved to South Africa, and I arrived in a country with a very pathologic history, 50 million people. 80% of people define as black South African, 10% white South African, 10% mixed ancestry, and a few 1%, around 1% Asian South African. There are 4 genetic medicine clinics. The one I used to work with was set by Peter Byton. He passed away about 10 days ago, peace to his soul. And this country, because of the pathologic history, have genetic conditions that align around ancestry. For example, if you are a white Afrikaners, the most common genetic condition is hypercholesterolemia. We also realized that because of the segregation, the country was the paradise of founder effect, and each single population group have condition that variant that uniquely are overrepresented. For example, in the black community, one specific variant is responsible for the Bardet-Biedl syndrome, and recently one specific variant in myosin 7A is the most common variant in Usher syndrome in black South African. Because of the environment of apartheid and the way people were treated, fetal alcohol syndrome became one of the most common conditions that we saw in our clinic. Surprisingly, we did not find sickle cell disease in black South African, to our surprise. Actually, most patients with sickle cell disease are migrant, mostly from Congo and West Africa. But sickle cell disease certainly was in South Africa some times ago. We studied the haplotype around the variant in black South African, and the sickle mutation probably disappeared because of the absence of malaria. Because there was no selective pressure, there was no need to maintain the variant in the population. So this is my son. He's 22 years now. That was 22 years ago in our apartment in Geneva. And when he was born, the umbilical cord below on the left-hand side is the true umbilical cord that linked my son to my beautiful wife. And I took part of that umbilical cord, moved back to the western part of my country, native country, Cameroon, and buried according to a specific ritual that we have to do to connect him to our ancestors. When I visited Mandela House in Soweto, when I was in South Africa, this is the house he used to live with Winnie Mandela. Behind the house, there is a tree, and under the tree, there is a voice of Winnie saying that we bury under this tree the placenta of all our children. So the exact same practices that we had in Cameroon. But remember, there is no sickle cell disease in Coastal South African. So that means that despite the fact that these two populations are from the same cultural extraction, their genetic composition is completely different because of the environment and evolving genetics within our population. So based on that, we put that the argument that we cannot consider a Bantu to be a Bantu from the perspective of genetics. That means that if we want precision medicine, for example, in that context, we need to speak of ancestry, but also from the geography where people come from. And based on that experience, we advocated at the time successfully with Max Mwanke, who was at the time the editor of the American Journal of Medical Genetics, to have a specific section for genetic morphology in diverse population. And that section had been very popular. So now I'll move to the main part of my talk, try to convince you that for the ancestry reason, for ecological reason, and for the equity reason, we all need to invest in African genomic variation. The first, the ancestry reason, we are all African. If you look on the left-hand side, the first guy is called Lee Berger. He's an anthropologist geneticist from Joburg. And the guy below him is the current president for South Africa. His name is Cyril Ramaphosa. And for a bizarre reason, they are involved in a kiss competition with homology. And homology is one of those archaic humanoids that evolved in South Africa that perhaps is one of our previous ancestors. in our genome today, and perhaps mostly in genome African population as shown on the graph on the right-hand side, the content of those archaic humanoid that we cannot yet read and that we call ghost DNA is about 5%. And we are not able to do it properly unless we study African population in a precise way. The data from my friend Sarah Tishkov, published a couple of months ago in Cell, suggests and propose at least 2 waves on integration of those archaic humans that never move out of Africa. But remember, some other archaic movement move out of Africa. And I guess Dr. Pabo got the Nobel Prize this year for medicine and physiology because he shows that part of the part of our DNA that is introgressed in our modern human was responsible for higher susceptibility to COVID-19 among European and Asian, partly. It's not the only reason, but partly. And the very same area on chromosome 3 have already been known to be associated with numerous phenotypes, including neuropsychiatric disorder. It was, he says, is true. That means that these archaic humanoids for which the DNA is yet to be properly read, what we call ghost DNA, should perhaps have some impact on disease and also on health. We know that because humans evolved in Africa at least 300,000 years and move out of Africa only 80,000 years, there are some variation that never move out of Africa. For example, this study, actually I have to revisit the study because long-read sequencing seems to indicate that the number on this study are not necessarily correct. But at the time it was published, it shows that 10% of the content of people with African ancestry in the Caribbean was not present in the reference genome. The data we reported from the History of Africa found 3.4 million variants not reported before. The data from Sarah Tishkov on 250 genome 2 months ago show 5 million variants not reported before. We know that even if you investigate besides single gene disorder, a complex condition like schizophrenia, your pickup rate is much higher in terms of association, a power of association in African population compared, for example, for a Swedish cohort, despite the fact that in this study there was only 1,000 Africans compared to 5,000 Swedish. We know that some of those variants that are very common in population of African ancestry may have impact on disease and also on specific phenotype. I think this is a cartoon boy of this illustration. This variant that is 200 times more present in Africans than in Europeans, associated with lower cholesterol at the 40% level, and associated now to the development of numerous medication against cholesterol. And most importantly, it's now been shown that you can actually gene edit this variant in adult and to modify the cholesterol expression. Some of the variants that we have in African population never move out of Africa can be associated with type 2 diabetes. So the first part, we are all African. Some variants never move out of Africa. We need to properly investigate them because we need to know our genome to properly implement genetic medicine. Africa have a north-south orientation, and this north-south orientation is associated with diverse climate. If you have diverse climate, you have diverse food. If you have diverse climate, you have diverse infection. And because of 300,000 years of history, there have been movement within Africa for those past 300,000 years. And because of this diverse climate, food of infection, who are a mode of natural selection, some variants in Africa have been selected. The story of sickle cell disease, selected against trypanosome, against malaria. The story of APOL1, selected against trypanosome. But we know that sickle cell disease is a serious disease, and APOL1 predisposed to kidney dysfunction in population with African ancestry. This is not the only history of selection in Africa. These are a chart that I borrowed from Sarah Tishkov, her recent work. In that study, she identified 65 other loci with natural selection. She investigated at the molecular level and physiological level at least 2 of those loci. So that means that we are yet to understand properly what are the extent of the natural selection that have evolved with the environment of Africa over the time. So I hope to have convinced you with the 2 parts that we all need to invest in Africa. I think that the Human Genome Project should have started 20 years ago with DNA from African population. I will now move to the third part of my presentation, and now to say, how do we invest in African genomic variation while maintaining the imperative of equity, specifically for population with African ancestry? Africa represents 15% of the world population, 5% of the world gross domestic product, and very few investment in research. So technically, we will say Africa do not have the capacity to do the research on his own DNA. It is not true, because we know that 30% of the world resources come out of Africa. There is just a asymmetry between the Africa wealth and the economy. So for that reason, people like myself or Aimé here, if you want to do your research properly, you need to go outside to look for funding. And this is represented, for example, on the chart. And the chart shows how we, based in Africa, we follow money, where the funding is either to Europe or the UK or to the United States. One of the problems of that is that when you only respond to call, some of the priority on the continent will not be addressed. For example, in this study we published more than 15 years ago, the hemoglobinopathy sickle cell disease will represent only about 5% of the study in genetics that are reported out of Africa. We will not have the opportunity to train people the way they should be trained. For example, we show that the level of knowledge for genetics for medical doctor and medical student were relatively low. The knowledge of genetics in the general population was not very proper. In that chart I showed before on Fragile X in the Western Cameroon, the population there think that people have Fragile X syndrome because it was a curse that was thrown by Chief K to the population. Chief K was a chief with 25 wives, and Chief K was a normal transmitting male for this condition, and Chief K, because of the 25 wives, all the granddaughter of Chief K was carrier for Fragile X, the reason why there is a kind of epidemic of Fragile X in this group. This is not theory, something that we have shown at the molecular level. We also know that working in Africa have not always been equitable in terms of collaboration. For example, 10 years ago, if you pull out the study published from Africa, you only have, for example, 30% of author that are Africans and only 14% of institutions even mentioned. But these have changed over the past 10 years. One project that have allowed the change is called H3 Africa. And H3 Africa have allowed to revise the precise genetic epidemiology or genetic structure of African population, and by filling a little bit some of the gap that we had on the PCA, allow African investigators to be currently present in the literature in the way they have not been before, allow African institutions to train the next generation on genetic scientists. This is a snapshot of people trained in my lab in Cape Town. Most of them are already doing their own career in various institutions across the world. So that means that tomorrow will certainly be different than yesterday. So I will use 2 areas or 3 areas of my research to show how we can address equity. And the first story I would like to tell tonight, I will call it the tragedy of the commons. I think I'm in the UK now. The tragedy of the commons is supposed to be in the books at the beginning, published about, I think, 100 years ago. But the tragedy today is the story of sickle cell disease, because it's still a very much neglected disease, described in America about 112 years ago. And one of the reasons I like sickle cell disease, I was mentored by great leaders in sickle cell disease. My immediate mentor, Stylianos, worked on sickle cell disease and described the haplotype. Stylianos was mentored by Hek Kazezian. Bizarrely, I'm occupying the office of Hek now. And he actually sent me that email. It's a real email of Hek, sent me just a couple of months before he passed, and to remind me that now that I was trained by Stylianos, I'm his scientific grandson. So it was so sweet. So the sickle cell is very common in people with African ancestry because of the selection to malaria. This is a condition that allows the distortion of red blood cell. And because of that distortion, they get stuck in the small vessel. They also get destroyed because of hemolysis. As a consequence, people with sickle cell disease will not have a long life if they live in Africa. For example, if you are born with sickle cell in Africa, you are not going to see your 5th birthday, 50% of children, if you are born in Nigeria. If you are born in America, you will see your 5th birthday because of newborn screening. But you still die early as shown on this map.
Over the past 40 years, the mortality rate in adult with sickle cell disease in the United States have not changed. They all die in the 5th decade or 6th decade. And the reason they all pass away is complication due to cardiovascular condition. They can be acute, they can be chronic. I will now convince you that if we invest in genetics in African population, we can alleviate that in 3 ways. The first way is primary prevention. Primary prevention is, for example, extending genetic diagnostic before birth, but we know we cannot extend that much because of ethical implication and the offering of the termination if possible. And we show that by doing some social science study across Africa, where clearly the attitude to termination, as shown on the right-hand side, was very different if you are a patient, if you are a doctor, or if you are a parent with one affected child. For example, a parent will have one affected child with sickle cell disease will accept the principle of terminating pregnancy because of sickle cell disease, which was not the case, for example, for doctors. We move focus to look for the way we can use genetic or genomic for secondary prevention. This is a chart I borrowed from Eric Green from the Genome Institute here, and where he shows that sickle cell disease, a complex trait, for example, on the right-hand side, are modified by both gene and environment. Sickle cell for monogenic disease are more simpler, but I would argue that sickle cell is both a monogenic disease as a complex disease. The reason is that if you have sickle cell disease and have variant in at least 3 genes, APOL1, alpha thalassemia, and HMOX1, those 3 variant in those 3 genes condition your capacity of having kidney dysfunction. And there are many other endophenotype sickle cell disease that are subjected to genetic variation. Lastly, we can use genomics to improve the treatment of sickle cell disease. We have known for at least 50 years that sickle cell disease patients who have high level of fetal hemoglobin will survive much longer. The quantitative trait that is fetal hemoglobin is subjected to genetic variation. Variation is at least 3 genes, including BCL11, allows some people with sickle cell to maintain a high level of fetal hemoglobin. If you remove that transcriptional repressor in a mice model, you cure the mice model with sickle cell disease. And when this was published by Stuart Hopkins, his group, we were all very excited and say, okay, we have the way to cure sickle cell disease. Until we have this series of patients published between the United States and Canada who actually has a deletion of BCL11 through a contiguous gene syndrome. They all have high level of fetal hemoglobin, but they also all have some level of intellectual disability and brain malformation. Fortunately, Dr. Ock in his group found a variation on an enhancer located 50 kb to BCL11 that can be modified and change the increase of the level of fetal hemoglobin only in red blood cell, and he called it erythroid-specific enhancer. And in other words, if you delete the transcriptional repressor, which is BCL11-1, you increase the level of fetal hemoglobin, but potentially with brain malformation. You change the variant in the enhancer a little bit before the genes, you increase the level of fetal hemoglobin potentially only in red blood cell. And this is the basis that allow the first gene editing of BCL11, successful gene editing of BCL11 to cure sickle cell disease. But I would argue that the BCL11 and MYB are not the only area in the genome that modify fetal hemoglobin. The reason is that our study on the African continent shows that those 3 areas only are associated to 20% variation of the level of fetal hemoglobin compared to 50% variation if you are of European descent. We know that because of the highest variation in African population, fine mapping is easier because haplotypes are smaller. And based on a African specific GWISE array developed by the H3 Africa Consortium at Illumina, we rerun the fetal hemoglobin genomic. I will show you the result a little bit later. Before that, let's look at the comparative analysis of current imputation panel using our data, our original data from sickle cell disease patients in Cameroon. On the first slide, on the first image on the left-hand side, this is the imputation accuracy according to the panel. The panel, the top med seems to be the most accurate, and it is normal because the top med panel have much more genomes. But if we look on the right-hand side on the amputation accuracy per chromosome, TopMed is still the beta panel, but we all see that most amputation panel on the that we have currently do not do well on chromosome 19. If we now look at the accuracy on the lower left side, and in that specific image, we look at the mean accuracy per chromosome and per panel, TopMed again perform much better. But in this analysis, we show that a custom panel that we developed based on 200 genome from Cameroon perform much better than the KPG panel. This specific analysis shows that local ancestry matters. And lastly, if you look at the number of variants across all the panel, those panels share only 1/3 of variant. It suggests that if you do an amputation on one panel and do not find an association, it doesn't mean that you have not found the association. You should do it with other panels. And based on that, when we analyze the GYs that we have from Cameroon, we use actually all the available panel. And I will show you just a glimpse of the result because we are writing on this story. For example, this is the discovery cohort. Of course, we replicated BCL11.1, which shows that our phenotype is correct. We replicated the MYB on 6, showing that our phenotype is correct. And then on chromosome 13, we found one novel association with fetal hemoglobin. And this is a true association because if you delete that gene in a mice model, the gene doesn't produce red blood cells anymore. And we extend our analysis to to the data available in the public domain, like the one in Tanzania, and also extend the analysis to the presently available data amongst African Americans with sickle cell disease. And we describe 14 neurocytes, and 7 of which are found in the meta-analysis, which clearly indicate that if you use an array that is African-specific, you're going to increase much more the discovery of neurocyte. Why do we need to do that? We need to do that because our patients are requesting it. This gentleman, his name is Grajevis. Grajevis is a Congolese patient. He lives with sickle cell disease. He's a graduate student at the University of Cape Town. He's a believer of gene editing. And these are my other cousin here, Tutankhamun. Tutankhamun died at 18. Tutankhamun, on the lower right-hand side, is the MRI of Tutankhamun bones. You will see that some of the bones, he has very short, he have an oligodactyly, like we call it in genetics, because he has some osteonecrosis of his bone. And these aplastic osteonecrosis are only seen in sickle cell disease. So many people think that he died of sickle cell disease. And if Tutankhamun had sickle cell disease, that means that for many, many years, many people like him have survived the age till reproductive age with sickle cell disease. That suggests that sickle cell disease may have put also some pressure on our genome. We have tested that hypothesis recently and found 3 areas subjected to natural selection only in people with sickle cell disease. And to push a little bit that study, we try to look in the variant in the exomes on people with sickle cell disease to see if there is an enrichment of some deleterious mutation in some area that you are not going to find in people without sickle cell disease. And we use 3 extremes. One group of people who survived much longer in Africa without treatment, so more than 50 years old in Africa without treatment. The other extreme were those who had stroke extremely at a young age, which is the negative extreme, and in between the two. And for example, in the longer survivor, we found a gene which is a calcium channel in which there is a recurrent mutation only in longer survivor. We also found increase and enriched variation in at least 3 genes associated with the metabolism of L-glutamine. We know that L-glutamine was approved 5 years ago as one of the treatments for sickle cell disease. It suggests that in this longer survival, they have some variation that allow them endogenously to produce L-glutamine. The story doesn't stop there. If we now look at those variants that evolved in Africa as a factor of selection, if you do a heat map of the prevalence of those variants across Africa, those variants seem to appear to exact same area. It suggests that a brave African like myself, I may have all the 3 variants in my blood. Each of the variant does something. For example, sickle cell can make you sick or protect you against malaria. APOL1 will make you have more susceptibility to kidney dysfunction. Alpha thalassemia will actually protect you against having early kidney dysfunction. What if you have the combination of all of those variants? So those are new areas where I think clever bioinformaticists and mathematicians will have to investigate in future for a sickle cell disease therapy. I think that what we should be doing is to be doing that through very large cohort that we can follow prospectively. I do think that we have the resources of doing now. We have a project funded by the NIH that allow us to follow a cohort of more than 30,000 patients across 7 different African countries. The second story I would like to talk tonight is the case of a silent epidemic. It's the story of hearing impairment. If in America, for example, 2 children out of 1,000 are born deaf. If you are in Nigeria, it's 6 out of 1,000, the same in South Africa. If a child is born deaf, usually in half of the case is because of a genetic origin. And for those that are genetic, most of the case is a hearing impairment without anything else. We call it syndromic. In Africa, we shown that we call it non-syndromic. In Africa, we shown that for non-syndromic hearing impairment, Waardenburg is the most common. Waardenburg is the association syndrome of hearing impairment and some pigmentation defect. For example, in those 2 SIBs, they have hearing impairment, but they also have striking blue eyes, very impressive on black skin. And so that means that actually you can diagnose Waardenburg in the street because having striking blue eyes is not the most common phenotype amongst people with African ancestry. What we have shown very early was that in Africa, the gene that is responsible for hearing impairment in half of the kids, European and Asian descent, are not common in Africa, in most African, GGB2. But we found one exception. In Ghana, we describe a founder variant responsible for about half of the kids that are born with hearing impairment. We showed that the founder variant actually evolved as a founder mutation by studying the haplotype around the variant. The same variant were found in a Japanese population, but in this study we showed that the haplotype in the Japanese, indicated by the red arrow, is very different from the one we found in the Ghanaian population. And our admixture mapping on the upper left-hand side clearly shows that the Japanese and the Ghanaian are located in a different place in terms of their location on that analysis. And we also probe the age of the mutation in Ghana and show by 2 different approaches represented on the right-hand side that the mutation evolved in a single Ghanaian ancestry about 10,000 years ago. So someone will ask, why is that some mutation evolved 10,000 a year and get fixed in the population? There is only 2 things. One thing is that it might be protect you against something. We went in that village in East Ghana and tried to ask for people. That village is actually called the deaf village. I think 1 of 2 people in that village have deafness. And we asked them, why do you think we have that much deafness in this village? And the most popular story is that there was a deaf man who was there for a very long time, long time. And he was very handsome. And he had a lot of kids with a lot of women because there was a lot of women after him. It's a funny story, but I don't think it's true. Because when we look at the skin of people who has that actual variant, if you have a double mutation with the variant, you have a thicker skin. So our current hypothesis is that the mutation gets fixed in the population, perhaps to protect against some kind of mosquito bite or kind of infection that was transmitted through mosquito. But of course, we still need to probe that properly. When we did the same study in Senegal, our first surprise was that we found a different founder mutation in Senegal. It was not the one we described in Ghana, but we also found the Ghanaian variant in Senegal. For those that do not know the geography of Africa, Ghana is very close to Nigeria. Senegal is far west of Africa, and they do not have borders. We look at the country around Ghana. For example, Ivory Coast, who share border with Ghana, do not have the Ghanaian variant in people with GGB2, even who are deaf. It's not the case in Nigeria. How is that the Ghanaian variant landed in Senegal? Our hypothesis is that the Ghanaian variant was brought to Senegal because Gore, where slaves were gathered before sent to America, was the shortest route to America. So if this is true, we should see the same variant among African Americans with hearing impairment. Actually, we saw it, and that's something I saw just 2 days ago. So we look at all of us, that who contain data, contain most people, most ethnicity in America. For example, the Ghanaian variant represented on the left-hand side, on the 101 allele that we found in America, 56% was among African Americans, and about 25% amongst Latin Americans who can have African ancestry. Absolutely inexistent amongst American European ancestry. So our surprise was the Senegalese variant. The Senegalese variant have the exact same pattern. So about 60% amongst African American, about 20% amongst African American with Latin ancestry. There was a third variant we described between Ghana and Senegal that was initially said to be in the literature to be a founder variant that evolved in the Mayan population in America, Native American, Indian American, from the Maya tribe. We check on that again using all of us. Actually, that Mayan variant that we found in West Africa is also very common in African American and in Latin American. I would suspect that that variant also arrived in America through transatlantic slave trade, and the first Mayan population that was investigated have some African ancestry. Of course, we have the tools now to investigate that. Actually, Noah, which is one of my graduate students at Hopkins, is investigating this because we have these data from both Africa, also now from all of us. But GDB2 is not the only variant involved in hearing impairment. We look at the other country in Africa for hearing impairment, and actually out of the 54 African countries, only 13 who actually have a single data published on hearing impairment. We know that at least 120 genes are associated with hearing impairment. If you look at those genes using a panel, the pickup rate is very low in Africans and also amongst African Americans. But there is a problem in these two studies. These two studies use single case, isolated cases. I think when we look at use the same approach for multiplex family, our pickup rate was much higher, 70%. But it suggests in both cases that novel genes are still to be discovered, and it suggests that Africa might be the place to discover those novel genes. Why do we need to think that novel genes has still to be discovered? If you look at the transcript of the inner ear, this study at least shows that at least 200 novel genes for hearing impairment are still to be discovered. The recent evaluation, for example, from Karen, still her work, suggests that 1,000 genes still need to be discovered. So we designed a study called Hygiene Africa for hearing impairment genetic studies in Africa. And we collected families segregating hearing impairment across 7 different African countries. And at the moment, of course, we have confirmed a lot of rare genes that was reported only once in one population. For example, for this gene variant was reported 15 years ago in an American family. We reported a second family, multiplex family in South Africa, confirming it as a new hearing impairment gene. We describe in details the profile of all the other gene variants. For example, this is the example of Ghana, showing a high level of genetic and allelic heterogeneity, a really, really high level of genetic allelic heterogeneity. And most importantly, we also describe a lot of novel genes. In this paper, for example, we put in 7 novel genes that we describe in Ghana. One person told me, why did you have to do that? We just had to do it because we needed to prove that if you investigate African population, you have a highly likelihood to have new genes. One of the genes, for example, PAX8, was in a family with Waardenburg syndrome. And fortunately for us, JAX already had a mice that was deleted for that gene, and the mice is actually deaf. The mice also was shown not to have thyroid, so we went back to the family and looked. So this is the depiction of the phenotype in the mice. When we realized that the mice did not develop thyroid, we went back to the family and measured thyroid hormone just to make sure that we are not missing anything. But fortunately, at least for those who were affected, the thyroid hormone was still okay. I'm not showing that it will remain okay because at least one member have a high TSH. In this other family, there was also a mice model with a malformation of the inner ear. But in 6 other families, there was no mice model. But we show, at least in silico analysis, shows that those genes were expressed in the inner ear. At the moment, one of our collaborators is developing 4 mice models for each of these genes. One of these genes is very interesting, POTEI, that we described in one of the family. It appeared to be a human-specific gene, so there is no mice autolog. One of my graduate students looked the other day at the copy number variation in that genes across the evolution, and we realized that the human have 4 copies of the genes, and it's only species who have that number of copies of the genes. That means that we suspect that it might be linked to language, because we are the only one who can speak a very complex way of communicating as compared to our nearest neighbor, the gorilla or the chimp. So is what we found in Ghana unique? No. These are the data I'm presenting for the very first time in public from Cameroon and Mali. We have the very same scenario. About 10% of new gene discovery, very high level of genetic heterogeneity, very high level of novel variants that have not been described. So 50% of the variants are not accounted for in the current database. We also explored the evolutionary genetics because we realized that we have some specific mice autolog in some of the variants, some specific gene without mice autolog. And this was a very simple experiment. Actually, we asked the intern to do it because we thought he's not going to find anything. We pick up from the very same population, people who are hearing impairment and people who are not hearing impairment. And we look in 150 genes associated with hearing impairment, derived and also ancestral allele, and we realized that actually there was a difference in allele frequency. There, it may mean 2 things. It may mean that even if you have one gene that explains hearing impairment, variant in the other genes may be associated to variable expression in the same individual. But it also means that some of the people with hearing impairment have enriched these genes for an evolutionary reason that we don't yet know and that we need to investigate. For this part of our story, we would like to say very few African populations have been invested for genetic of hearing impairment. The whole exam study that we are presented now show a very high level hill for resolution, 90%. There's still a high hill for gene discovery, about 10 to 15% if you study African population. Comparatively, for those that are in the field, if you do the same study in people with European ancestry or Asian ancestry, the rate of discovery new genes now is 1%. So you need 100 families to have one new gene discovery. Because there is a high level of genetic heterogeneity, but mostly allelic heterogeneity, we think that studying African population will improve our hearing impairment gene pair curation. As we generate this data, there will be some ethical questions that will evolve. One of them is the actionable genes. Should we return or not? There are three reasons to return to investigate it. One is scientific. There is no reference data. Most of the variants that we found are novel. How do we handle that? The second is ethical. Because we've seen these people, what do we do with the genes variant? The other is practical. Who should return? We don't have genetic counselor much in Africa. So we have a group of people. It's a funded NIH grant. We are trying to look at those questions. While waiting to having an answer, we know that what we have generated here will assist to improve the database. We know that it's an imperative also because without that element in the database, we're going to make a mistake to infer deleteriousness as shown in this data for cardiomyopathy. So I will argue that this big building that was proposed by Dr. Collins at the next step for the Human Genome Project still have a few cracks in the foundation, the missing link in African genomic variation. The reason I proposed a couple of years ago that to address those links, we need to sequence at least 3 million Africans. And if we don't do that, we will be shooting a foot on our foot, but most importantly, we will not be able to make genetic medicine equitable, innovative, and sustainable for all population. I would like to thank you for your kind attention.
Thank you very much.
Thank you.
Professor Ambroise Wonkam. I think we already have questions online. Otherwise, I would start with one question of mine. When we were growing up in Africa, we had this book to learn English. The book was called English for Africa. Now you have talked about research, genomic research in Africa. How would you translate this into genomic research for Africa in terms of benefit sharing with the African research participants?
First of all, I did not read the same book. Because when I grew up, I was not even speaking French. I was speaking my mother tongue. I started speaking some French only at 5 years, and the English, which is still broken, probably around my 18. So the way I see it, I think there are two ways of saying it. The way you have asked the question is on the angle of equity. How can we make genetic useful for Africa? I think I argue usually with funders on the angle of the scientific imperative. We are all African, so that the first precedent. And because we are all African, we want to know what happened before our brother and sister moved 80,000 years ago to populate Europe and Asia. And that's critical, because if we do not have that information, some of the missing link on our understanding of the human genome will not work. So that for everyone else. So to come back home now, how should you and I do? I think we should have to continue to do what we do best, to talk to this meeting, advocating to people like Les, to train as much people as possible, to train them efficiently in such a way that they can read that very old genome of the human family. I'm not talking just training doctors, training counselors, training nurses, training computer scientists, training science communicator, also training policymaker, because actually when you approach people who take decisions, sometimes the best decision is around a coffin in a garden. Someone just moves some numbers in an Excel sheet and you have a program that is funded. So those things have to happen at all those levels. And it's the only way I can put it that way. So there are many, many layers in which we need to invest to make genetic medicine equitable in Africa and beyond Africa.
Thank you.
All right, ready for the questions?
Yes.
Great. Welcome questions in person, and we do have, where are our mic runners? Do we have some mic runners? Yes. All right, so please put your hand up. We have, looks like 3 mic runners, if you're willing to ask a question. I'll start. 3 million.
Yes.
So Africa genomically is a very complicated continent. How are you going to distribute those 3 million across the continent, and what percentage of the diversity are you projecting that you can capture with 3 million?
Okay, thanks, Les. I think when we proposed the idea of 3 million, and the question still came the other day from one of the editors of Nature, why 3 million? When the first 4 colleagues who described the Human Genome Project, they shows that every 1,300 nucleotides, I'm different from him, different from you, because of single nucleotide variation. So that means that for the now 3 billion nucleotides we have, we are different. 9 billion nucleotides we have, we are different with 3 million at least single nucleotide variation. So if you imagine that each African, because of the complexity, have at least one variation that is different from anyone else in the reference genome, you need minimum 3 million people. It's a gross calculation. So how do you split the 3 million people? Let's pick sickle cell disease as a single variant. We say sickle cell disease evolved in Africa. I think the best estimate now say about 10,000 years ago. But we have sickle cell disease in people in Italia. We have sickle cell disease in people in India. We have sickle cell disease in Middle Eastern population. So how is that young person who has the first mutation arrived there? He should have crossed the desert. He should have swum in the Mediterranean. At that time, there was no plane. And he also had landed the other side, right? In each of the part of his journey, he admixed with local population. He also changed the content of the DNA because the environment in the desert is not the same like in the Mediterranean, and it's not the same certainly like in southern part of Europe or in Greece. I'm here putting the concept not only of the origin, but also of the geography. So we will need to split Africa in little portions based on ethno-linguistic, but not only on ethno-linguistic because the Bantu in South Africa are not like the Bantu in Congo. In some places where there is a high genetic diversity, and we can use that, we can do that using principal component, we may have to substructure within some country. For example, in Sudan and in Cameroon, you have much more diversity than anyone else in Africa. For example, in Nigeria, there is a tribe called the Boron population who seems to have a genetic composition which is much closer to people in the eastern part of Africa from Ethiopia. So we also need to rely on the anthropology, local anthropology, people talking their story, and to know how we sample. So it has to be per country, it has to be per region, it has to be per linguistic, but also it has to be per the story that people will tell you. How are we for that? Are we close to do the 3 million? We are not yet close, but we are close to launch the project. At the moment, the project will have a name that is called GENCO, Genomic Center of Excellence in Africa. At the moment, the leadership of the project is 3 people from Africa, myself, Christian Happy from Nigeria, and also Nicola Mulder from South Africa. We have a group who is advocating strongly for us. That group is made of 3 people. The one is Francis Collins, of course, the other is Charles Ochimi, and also we have John Kengerson and a few other colleagues. So we will be presenting an event on GENCO at the UN Science Summit on the 17th in September in New York. And we will organize that event jointly with the European Union, of course, and the African Union. The plan at the moment is to set the coordinating center of GENCO in Ethiopia with a direct access to the African Union through CDC Africa. That will allow us to have a strong support from the government. We have some commitment from the industry already, organized around a group called Praga. We have needed support from the White House at the moment. We are working toward launching the project sometime next year.
Fantastic. Questions from the audience? You were talking about the challenges of returning results in Africa. And one of the limitations you mentioned was, to paraphrase you, I think, is an inadequate base of healthcare providers.
Yes.
What is your strategic thinking about genetics, the provision of genetic healthcare, genomic healthcare in Africa? Meredith will probably kill me for saying this, but I have to say in the States, the profession of medical genetics is a double-edged sword. It's our greatest source of expertise in medical genetics, but it's also extremely limited in number and penetration within healthcare. So how are you thinking that the medicine of genetics should be deployed in Africa?
So I think there are many layers to your question, Les. The first principle is that if genetics is important in America, it should be important everywhere else. If genetic services are available in Europe, it should be available everywhere else. But how do you strategize on it? I'll pick up maybe the first arm of your question, actionability and the return of result. When we look at the actionability from the angle, for example, of the American College of Genetic Medicine, the way it started initially was a list of 56 genes, right? And those 56 genes, if you look at the structure of those genes, it's nearly only 2 types of condition. The one is inheritable cancer. The second group is inheritable cardiovascular condition. I think the list from the UK was more pragmatic. It was a limitation of about 10 of those genes. So the first thing is that is it the right approach? How we know? Because you need to know also the burden of disease in that population. Is the right approach only looking for single gene disorder, I will argue again no. So let's move those two approaches in Africa. So if we want to return in Africa, we add sickle cell number 1. We add APOL1, which is not a single gene disorder, but a predisposition disease, which is important. I will even actually add sickle cell carrier as an actionable, because actionability will not be only limited to providing specific care, but also to prevention, and in which case a sickle cell carrier has to be part of it. We will add one specific variant that allows the metabolism of a specific drug for HIV, which is very much used in Africa. So actionability will have to extend for variant in pharmacogenomics. So how do you now design a program, a genetic program for Africa? I'll just tell you one story. I remember when I came back in Cameroon, first I had some good connection, and I went to see the Minister of Health and told him, I'm a geneticist, I want to do a genetic service. He said, oh, why are you crazy? It's not my priority. I have some malaria and an HIV issue here. You say you're going to want to do a genetic to do what? It was about that. And I asked Your Excellence, that's the way you say it. How do you handle pregnant women who are HIV positive? And he tell a long story to say that they will use a Western blot approach to test if the baby is affected. And I told him, if you provide me a lab, I will tell you if that baby is affected in 2 days, because I will use copy number of the virus to know if it's affected or not. He turned to someone and asked, is that true? And the person came back after a couple of hours, say it's true. That's how we had our first genetic lab. So we use investment from infectious disease to actually set a genetic lab for human disease. What I'm trying to underline here is that in the context of Africa, you cannot approach it strategically in the same way you approach it in Europe. You need to use the burden of the disease locally and to see how you creatively use the resources that are linked to the burden of disease to develop a genetic medicine services. So that at the policy and strategy side. But at the training side, you need to do the same everywhere. I think you need to have program for human genetics. You need to have program for genetic medicine. But for program for genetic medicine, I'll put a little bit of bemol, because if you want to do it in a way I learned to do it in Europe, it will not work. You better start with pediatricians who see a lot of geneticists and have like some short training to make sure that they are kind of aware of what is happening. You need to start with nurses in community centers and have some short training in genetic medicine, such because they are the point of entry. I'm a geneticist, medical geneticist, but I've never seen anyone anywhere that woke up in the morning and say, today I have an appointment with my geneticist. People say, I have an appointment with my GP, I have an appointment with my gynae, with my pediatrician, but no one say to my geneticist. Mm-hmm. People land up to our offices through another specialist who refer it. Mm-hmm. So your best advocate will not be even the GP, it has to be the nurse. So I will design a project that empower that first line. And lastly, for Africa, I will put the emphasis of hemoglobinopathy. In the last 10 years, there was some concept in the press called the genetic bubble that we all faces at the time. People will say, you provide us too much hope, but there's no delivery. That bubble will not happen in Africa, because if you set only a simple hemoglobinopathy clinic, you're going to have so many patients to occupy your whole life.
Any last questions? I think we're about out of time, so you want to keep going?
We have a break. Yeah.
Oh, question. Microphones, please. Down front here.
Professor, I listened that you are working with SOX9 and the deaf partners, and we're trying if you are also searching in the DSD patients.
Yes, that's correct. SOX9 definitively is part of the genes involved in GSD. But I would tend to think those families in which we found SOX9, they have a apparently non-syndromic hearing impairment. It's involved not only with this GSD, also involved in skeletal dysplasia. In some other syndromes, SOX9 will be associated with a GSD plus short stature. It wasn't the case in that family. We tend to suggest that perhaps SOX9 perhaps have some level of pleiotropy in terms of expression, so that we haven't yet properly investigated. Having said that, we have a second family with SOX9 from South Africa this year. We will see if the family in South Africa have the same clinical expression than the one we found, like the one we found in Ghana.
Thank you.
Hi, Ambroise. Thank you for a lovely talk. My first question was sort of leading on from the previous discussion about how you start to implement and embed genetics in Africa. And I wondered if you'd done any sort of engagement of prescribers at all, because that could be another route as well for helping sort of implement pharmacogenetics and, you know, bringing the benefit of genetics in better prescribing and better outcomes for patients. It's quite more of an immediate sell, as it were.
Yeah, I think you're correct. I think I want to be realistic here. Let's say even if we pick the setting where we are now in the UK, the people who actually use pharmacogenomics in clinical practice is not that much. Even for those areas where we have a true proof of principle, for example, pharmacogenomic and clotting disorder, and it's not that in all genetic clinic where people have the reflex, even though there have been some very solid proof of principle. I would guess that in Africa, perhaps you should use a different group of pathology. And I think HIV is the one that we can use as a proof of principle for pharmacogenomic being used in clinical practice. But I still think that for the policy perspective, the most common genetic condition of like hemoglobinopathy of sickle cell needs to be the way to start. Yeah. I think it needs to be the first place to start. Because even in sickle cell disease and hemoglobinopathy, you can actually still use pharmacogenomics. We know, for example, that the medication that seems to work, which is hydroxyurea, associated with increased level of fetal hemoglobin and lowering of white blood cell in patients, doesn't work for all patients at all. Actually, 25% of patients seems to be resistant. That's the first place to start the pharmacogenomic study. And when you convince people with that specific case scenario, because we all know at least one person who passed away for sickle cell disease, a friend, a family member, a cousin, or whoever, you cannot extend. And the second place would probably be HIV treatment.
So thank you very much for the questions. And we thank you, Ambroise, for your keynote presentation. So Ambroise is still with us today. You can still approach him during the breaks and ask questions. And for now, we have a very quick comfort break because we need to come back in 5 minutes for the first session. We are already running a little bit late. So thank you very much.